So, students, in the last class, we have discussed about amino acids and the classification of amino acids and their function. So, in this lecture, this is the second lecture in a module in the proteins and amino acids and proteins module. And this module, we will be discussing about the peptides and the peptide bond formation and the physiological important peptides. Then we will be discussing about the classification of proteins and their functions. Then the organization of the protein structure, that is the primary, secondary, tertiary, quaternary structure. And finally, I will just give you a overview of the Ramachandran diagram, which is important to understand the structure of the protein. So we have already discussed about the amino acids and their classification and the titration curves and so on. And now coming to the peptides, peptides consist of two or more amino acid residues. They are linked by the peptide bond. You can also see here, this is one amino acid and this is another amino acid. These two amino acids are linked head to tail to head fashion. So that means the carboxyl group of the one amino acid and the amino group of another amino acid are involved, are connected by the peptide bond, and this peptide bond is nothing but the C double bond or NH bond, and this peptide bond formation is always accompanied by a loss of a water molecule. You can see here when the two amino acids are condensed, the water molecule is eliminated and the peptide bond is formed. You can also see here, and this is the representation of the Chitraionic form of the amino acid, and it is the normal form of the amino acids. And so that means if there are four, if the peptide has the four amino acids, for example, this is one amino acid and this is the tyrosine and the aspartic acid and the glycine, and if the four amino acids are connected, and we would see the three peptide bonds. That means if the four amino acids are there in a peptide, the three peptide bonds are present. That means always the number of amino acids minus one would be the number of peptide bonds present. And these peptide, and of course you can see here it is a pentapeptide, and it has the four peptide bonds. You can see here, and the peptide bond has the double bond character. So in the sense, what the peptide bond, what we are seeing here is that it is neither single bond nor complete double bond. It has the partial double bond character because of the resonance of the carbonyl pi electrons and the lone pair present on the amide nitrogen that gives the peptide bond partial double bond character. This we will explain later because of the resonance. You can also see here when this is the peptide bond, this is the remaining part of the protein, and this is one peptide bond is highlighted here. And when the carbonyl pi electrons migrate on to the oxygen, and the oxygen gets a negative charge, and the carbon gets a positive charge, and when the lone pair of electrons contribute for the bond formation, again you would the bond is formed between the carbon and the nitrogen. So that means the double bond character of the peptide is because of the resonance of the carbonyl pi electrons and the amide nitrogen. That's why the peptide bond has the partial double bond character. And so now there are different types of peptides. Like if the two amino acids are present in a peptide, then it is known as the dipeptide. If it is three, tripeptide, four, and and so on. And the dipeptide, we have the two important dipeptides present in the muscle and the brain. One is the carnosine, second one is the anserin. And you can see here the carnosine has the beta alanine and histidine, whereas the anserin has the beta alanine N-methyl histidine. That means these two are composed of the two amino acids, and this carnosine has the important, it acts as ideal buffer in the muscle tissue, and in addition to this, it has many functions and antioxidant, anti-glycation, metal chelation, and so on. And in the same way, anserin also, as I said, that it is composed of the beta alanine N-methyl histidine, and this anserin also has some important functions. It protects against fatigue, and it also has antioxidant function. So coming to the aspartame. Aspartame, we know that it is the sweetening agent. It is composed of the aspartyl phenylalanine. You can see here, this is phenylalanine, and this is the aspartic acid, and it is nothing but the aspartame, and it is present in the African berry. It is a sweetening agent. So coming to the tripeptide. Tripeptide consists of three amino acids, and these three amino acids are connected by the two peptide bonds, and we have important tripeptide, that is the glutathione. You can see here the glutathione consists of glutamic acid, cysteine, and the glycine, and it is also known as the gamma glutamyl cysteine glycine, because the gamma carboxylic group is involved in the peptide bond formation instead of the alpha carboxylic group. That's why this is the gamma glutamyl peptide cysteine. So in any amino acid, we notice that this carboxyl group supposed to be involved or supposed to link with the another amino acid with peptide bond, but in this case, the gamma carboxyl group, that side chain carboxyl group, is involved in the peptide bond formation. That's why the glutathione is also known as the gamma glutamyl peptide cysteine glycine. This is cysteine, and this is the glycine, and the glutathione has many functions. It acts as a reducing agent in the cell, and it assumes the dimeric form during oxidation. You can also see when it is oxidized. This is the GSH this is the reduced form. When it is oxidized, it forms the GSSG. That is, this is the oxidized form of the glutathione, and this is responsible for keeping the cell in the reduced state, and it participates in many reactions, that is, such as the removal of the hydrogen peroxide in the erythrocytes, and removal of the toxins, and release of hormones, and it also protects the body from the radiation, and it regulates the telomerase activity, and it also involved in the modulation of apoptosis. So this is the non-enzymatic antioxidant present in the human body, and this glutathione plays a very important role, and it is also cofactor for many enzymes. Even if you can see here, glutathione peroxidase also uses glutathione as a cofactor, and this glutathione peroxidase is involved in the conversion of hydrogen peroxide to the water, and of course, this hydrogen peroxide is formed from the superoxide anion by the superoxide dismutase, and so this is about the glutathione. Then coming to the thyrotropin releasing hormone. This thyrotropin releasing hormone consists of three amino acids, that is glutamic acid and the histidine, as well as the proline, and it is mainly secreted by hypothalamus, and it stimulates the release of the TSH and also the prolactin from the anterior pituitary. You can also see here. This is the thyrotropin releasing hormone. This which in turn triggers the it is secreted from the hypothalamus, and it also stimulates the release of TSH and prolactin from the anterior pituitary. And then the chemotactic peptide, and the chemotactic peptide consists of formal methionine, leucine, and phenylalanine, and it is present in the leukocyte, and it plays an important role in the chemotaxis. And then coming to the enkephalin, it is a pentapeptide. It is composed of tyrosine, glycine, phenylalanine, methionine, and this enkephalin is present in the brain, and it functions as the body's own opiate or the analgesic. And then we have angiotensin 2 and the angiotensin, and this angiotensin 2 is a octapeptide. You can see here, this is formed from the angiotensin 1, which is in turn formed from the angiotensinogen, and it is a part of the angiotensin renin system. And this angiotensin 2 is a octapeptide, and you can see here, this is octapeptide, and it is formed from the angiotensinogen. And the angiotensinogen, by the action of the renin, angiotensin 1 is formed. Angiotensin 1, by the action of the angiotensin converting enzyme, forms the angiotensin 2. This angiotensin 2 is a octapeptide, and it is a powerful vasoconstrictor that raises the blood pressure, and so on. And then coming to the bradykinin. So bradykinin is also a nonapeptide, and it has the 9 amino acids. but it is found in the lungs and other cells, and it is a powerful vasodilator, and it lowers the blood pressure. You can see angiotensin, it is a vasoconstrictor, whereas the bradykinin, it is a vasodilator. And then the oxytocin, we know this oxytocin and vasopressin, these are all sister hormones, and this oxytocin is also the nonapeptide, and this oxytocin is mainly synthesized in the hypothalamus, and it stimulates the uterus contraction, and it is also secreted from few other tissues such as the ovaries and testes, and it causes the stimulation of the ejection of milk. And then coming to the vasopressin, it is also nonapeptide, and you can also see here, and it has the disulfide bridge between two cysteine residues, and it is also called as the antidiuretic hormone. It is mainly involved in the regulating the body's water, or that is the retention of water. So that means what is the conclusion from here is that all these peptides have either 2 or 3 or 4, 5, 8 or 9 amino acids, but they have various functions in the human body. And coming to the proteins, we have the protein, we have already explained in the first module that the proteins, the word protein is derived from a Greek word that is proteus, meaning that holding the first place or the first rank in the living matter. So that is the meaning of the proteins, and we have already said that the proteins have many functions. They have the structural role, they have the defense mechanism, transport function, contractile function, and some proteins are hormones. There are some proteins are some toxins are also the proteins in nature. So this we have already explained in detail in the first module. We can go through this later. So that means the proteins have various functions in the human body, and these proteins are these proteins are composed of many amino acid residues linked by the peptide bonds, and they have certain special properties. They have the high molecular weight, and they have the colloidal nature, and they have different properties, and they are soluble in different solvents, and they have the different shape, and so on. Then coming to the charge properties of proteins, we have already discussed the amino acid titration curve, where we have already clearly mentioned that the charge on the amino acid is dictated by the pH. So we have already clearly seen that when we titrate any amino acid with alkali, how the charge changes. In the same way, the charge of the protein also depends on the surrounding medium. So this is the protein, and when you're depending upon the pH of the medium, the charge is dictated by the pH of the surroundings. And so that's why this property is also used to separate the proteins. Charge properties of proteins is also used to separate the proteins by ion exchange chromatography. I think this you must have already studied in the analytical course. And coming to the isoelectric point, so all proteins have isoelectric point in the sense the pH at which the net charge on the protein is 0, and every proteins have the characteristic isoelectric point. So at isoelectric point, the protein has the protein has a minimum solubility, and that means the protein is almost insoluble. That's why this at the isoelectric point, the protein, any protein, when it is present in the solution, when we adjust the pH of the that solution to the isoelectric point, the protein tends to exhibit the least solubility. As a result, that protein can be precipitated from the remaining proteins. So different proteins have the different pA values, and this is very, very important to understand, in the sense that the pA value, as I already said, that the pH at which the net charge on the protein is 0 is the isoelectric point. And you can see here some proteins have the higher pA value and some proteins have the lower pA value, and this pA value is dictated by the amino acid composition of the protein. And so that means the protein has more of basic amino acids in its structure, and its pA value lies on the alkaline side, and if it has more of acidic amino acid, its pA value lies on the acidic side, and so on. So therefore, the pA value depends on the composition of the amino acids present in a polypeptide chain. And you can also see here, as I already said, that any protein, if you take at the isoelectric point, the solubility, this is the y-axis is solubility, x-axis is the pH. At the isoelectric point, the protein has the least solubility because it has no net charge is 0, so it cannot no more interact with the surrounding water molecules in order to make it more soluble. And so that's why, so every protein has its particular pH value. And since the proteins are the amphoteric substances, because the protein has both acidic amino acid, basic amino acid, many, and so they also acts as a buffers. For example, hemoglobin present in the erythrocytes and also plasma proteins are important buffers in the human body, and they regulate this. You will understand how the hemoglobin regulates the pH in the next lecture when we, after the exam, when we take the detailed structure of the hemoglobin and the myoglobin, why the hemoglobin is a transport protein, why myoglobin is a oxygen storage protein. This we will understand. There you will understand the importance of the hemoglobin. It is not only transporting the oxygen, it is also regulates the pH of the blood. That time we will discuss. So this is about the general properties of the proteins. So coming to the classification of proteins, and the proteins are classified in various ways based on their composition, based on their solubility, based on their shape, and based on their function. So they are classified in various ways. So based on their composition, the proteins are classified into simple proteins, conjugated proteins, and the derived proteins. So in simple proteins, in the sense they are composed of, they are built with only amino acid. That means when the simple protein is hydrolyzed, only we get amino acids. So that means they are made up of with only amino acids. For example, human plasma albumin, trypsin, chymotrypsin, all these are simple proteins. They have only amino acid residues. You can see here albumin, globulin, all these are the simple proteins. And coming to the conjugated proteins, and one more point is that most of the simple proteins are globular in nature, and in the sense they don't have any non-protein component attached. They are only built with the amino acids. And coming to the conjugated proteins, these conjugated proteins are composed of simple globular proteins and also with the non-protein component. That means when the conjugated protein is hydrolyzed, we get the amino acids and also the non-protein component, and that means conjugated proteins are composed of the protein part as well as the non-protein component, and this non-protein component is also known as the prosthetic group. And so therefore, depending upon the prosthetic group, if the prosthetic group is lipid, then they are known as the lipoproteins. If the prosthetic group is carbohydrates, then they are known as the glycoproteins. If the prosthetic group is phosphorus, phosphoproteins. If the prosthetic group is nucleic acids, nucleoproteins, and so on. So that means the non-protein component present in the conjugated proteins is known as the prosthetic groups. And the third one is the third class of is the derived proteins, and these derived proteins are mainly the degradation products obtained from the normal proteins, either by the hydrolysis or by the coagulation by heat or the alcohol. So these derived proteins are again further classified into two types. One is a primary derived protein. Second one is a secondary derived proteins. So primary derived proteins, in the sense when the protein is subjected to the heat or the alcohol treatment, the protein loses its three-dimensional structure. As a result, we get the primary derived protein, but the peptide bonds are intact. Whereas the secondary derived protein and the secondary derived protein, when the protein is subjected to the partial hydrolysis, and the secondary derived proteins are formed, and the proteases, peptone, gelatin, all these are secondary derived proteins. And in the secondary derived proteins, the peptide bond is also the peptide bond is also cleaved because of the hydrolysis. And this is about the one way of classification. And another way of classification is based on their solubility. Proteins are also classified into albumins, globulins, glutamines, protamines, histones, prolamines, and scleroproteins, and they have the different solubility in different solvents and the different functions. And other classification is the based on their shape. So this we have already seen. We have already said even in the first module that the proteins are classified based on the shape. They are classified into two categories. One is the fibrous proteins. Second one is the globular proteins. When we say globular proteins, they have the globular shape. You can see here. So globular structure, and they are soluble in the water and also the aqueous solutions of acids and bases. And these globular proteins fold into a variety of fold into spherical shapes, and they have various functions. Usually, these globular proteins function as enzymes, or the hormones, or the transport proteins, and so on. For example, you can see here hemoglobin, myoglobin, albumin, lysozyme, chymotrypsin. All these are globulins. And coming to the fibrous proteins, the fibrous proteins you can also see here. They have the elongated or the extended structure, and for example, collagen and keratin. And these fibrous proteins have the long thread-like structure, and they have mainly the structural role in many animal tissues. For example, the alpha keratin, beta keratin, collagen, and elastin. All these are the fibrous proteins. That means based on the shape, their proteins are classified into globular and the fibrous. Globular proteins have various functions, like they act as enzymes, hormones, and so on. And the fibrous proteins have elongated, extended structure, and they are mainly the components in the structural material of animal tissues. And this is about the classification of proteins based on their shape. So coming to the protein structure, and we have already we have already explained in the first module that the structure, the function of the protein is mainly dictated by the structure of the protein. And so therefore, since the proteins, as we already said, that they are made up of with several amino acid residues, they are connected by the peptide bonds, and such a linear sequence of the amino acids and such a linear arrangement is highly unstable, and this linear arrangement tends to fold into a particular shape, and this is known as the conformation. And there are many stages are involved in the formation of the three-dimensional or the functional protein from the polypeptide chain. And so in order to understand these stages, the protein organization is divided into four levels. That is, one is the primary structure, and second is the secondary structure, third one is the tertiary structure, and the quaternary structure, and so on. And so in brief, the primary structure means primary structure refers to the sequence of amino acids present in a polypeptide chain, and the secondary structure refers to the folding of the secondary structure refers to the folding of the linear polypeptide chain, and the tertiary structure refers to the folding of the polypeptide chain influenced by the R groups into the three-dimensional shape, and the quaternary structure refers to the association of one or more polypeptide, one or more subunits. And we know that hemoglobin hemoglobin is a tetramer. It exists in the quaternary structure. So we will briefly see the what is the primary, secondary, tertiary, and quaternary structure in detail. And as I already said, that the primary structure refers to the sequence of amino acids present in a polypeptide chain, and this primary structure is stabilized by the peptide bond present between the amino acid residues. And in case of insulin, the primary structure is also stabilized by the interchain and the intrachain disulfide bonds, because insulin doesn't fold into three-dimensional shape, and it is present. It has two polypeptide chains, and A chain and the B chain, and these two chains are connected by two interchain disulfide bridges. And in addition to this, one intrachain disulfide bridge present within the A chain, and these disulfide bridges are very, very important for the insulin to function. So that means the primary structure is mainly stabilized by the peptide bond, and it is nothing but the sequence of amino acid residues present in a polypeptide chain, and it is mainly stabilized by the peptide bond. In case of insulin, it is also disulfide bonds also plays an important role in the stabilization. And of course, different proteins have the different number of amino acids. You can see here it is given, and also the number of subunits and molecular weight. And this is about the primary structure. Second one is the secondary structure. The secondary structure refers to the folding of a polypeptide chain along the axis. As I already said, that the primary structure, that is the sequence of amino acids in a polypeptide chain, amino terminal to the carboxyl if it has the 1,000 amino acids, such a lengthy polypeptide chain is highly unstable, and it tends to fold either orderly or in the orderly or in a disordered manner, and in order to form the particular conformation. So therefore, the folding of this polypeptide chain is along its axis is known as the secondary structure. And if the polypeptide chain folds in a ordered fashion, then the ordered secondary structures are formed. It is also known as the ordered conformation. If the polypeptide chain folds randomly, and the random secondary structure or the disordered secondary structures are formed. And we have two ordered secondary structures. One is alpha helix, and second one is a beta platelet sheet. And we know that in alpha helix, in the alpha helix, you can see here, this is the alpha helix, and in the alpha helix has a certain characteristic features. In the sense, in this the polypeptide chain is tightly coiled along the axis, and therefore these coils are highly stabilized by the intrachain hydrogen bonds. You can see here what you are seeing here. These are the intrachain hydrogen bonds, and if you see the alpha helix in this when it is coiling, and the distance between the two amino acid residues in this coil is 1.5 angstroms, and each turn has the 3.6 amino acid residues, and the R groups is always you can see here, the R groups is always projected outwards. You can also notice here the R groups are always present, that side chains are always projected outwards, and the pitch of the alpha helix is 5.4 angstrom. The pitch is nothing but the distance between the two equal points on the helix, that is the pitch, and the pitch is 5.4 angstroms, and the width is 5 angstroms. And this alpha helix is stabilized by the intrachain hydrogen bonding, and in this the hydrogen bonds are formed between the NH group of the first amino acid and the carbonyl oxygen of the fourth amino acid. Every carbonyl oxygen and the amide hydrogen, the first and the fourth are involved in the hydrogen bond formation. So that means every carbonyl oxygen and the amide hydrogen are linked by the intrachain hydrogen bonds, and that means the carbonyl oxygen of the first amino acid and the amide hydrogen of the fourth amino acid are involved in hydrogen bonding. In the same way, the carbonyl oxygen of the second amino acid, then the amide hydrogen of the fifth amino acid are involved in the hydrogen. That means every first amino acid and fourth, second and fifth and third and sixth, they are involved in the hydrogen bonding, that is the intrachain hydrogen bonding. That's why the intrachain hydrogen bonding gives the maximum stability to the alpha helix. You can see here the extents of hydrogen bonding. Every carbonyl oxygen and the amide hydrogen are involved in hydrogen bonding. So this hydrogen bonding gives rigidity to the alpha helix and also to the stability to the alpha helix. And as I already said, that the polypeptide chain can fold into either right-handed or the left-handed, and the right-handed alpha helix are more stable compared to the left-handed. Left-handed alpha helix is unstable because of the steric hindrance by the side chains, and the right-handed alpha helix is more stable than the left-handed alpha helix. And in this alpha helix, some amino acids can stabilize the alpha helix, some amino acids can destabilize the alpha helix, and especially the proline acts as a helix breaker or the helix terminator. And some amino acids can stabilize alpha helix, some amino acids can destabilize alpha helix, and some proteins like keratin and collagen are almost it has the alpha helix. And so this is, you can also notice here that when it is folded into the ordered form into alpha helix, you can see here every carbonyl oxygen and the amide hydrogen of the first and fourth are involved in the intrachain hydrogen bond formation. That's why this intrachain hydrogen bond formation gives the rigidity to the structure of the alpha helix. And you can also notice here clearly that every carbonyl oxygen and the amide hydrogen involved in the hydrogen bond formation. And of course, depending upon the, for example, usually if the alpha helix is any alpha helix is in the proteins, the alpha helix has different types of amino acid. But for example, if the alpha helix is made up with only one amino acid, like it is hypothetical polypeptide, assume that the hypothetical polypeptide, if it is only made up with leucine, only made up with alanine, are only made up of with glutamic acid or aspartic acid. Depending upon the pH, either they can form the random structures or the ordered helical structures, because as we have already said that the each and every amino acid has a different PA value and the different pKa values of the alpha carboxyl group and the alpha amino group, as well as the side chain group, and depending upon the when it is present in a polypeptide chain, depending upon the pH and the side chain functional group, either it is protonated or the deprotonated. As a result, either it can form the ordered structures or the random helices. And in addition to this, alpha helix, the alpha helixes are again different types. 310 helix, in the sense it has the 3 residues per turn and the 10 atoms, and of course then pi helix, it has the 4.4 residues and the 16 atoms, and this is not that much important. Then coming to the second, the ordered second structure is the beta platelet sheet, and we know that these beta platelet sheets are they have the sheet-like structures. You can see here they have the sheet-like structures. They are formed when the two or more polypeptide chains are lined up side by side, and the beta platelet sheets are formed. And these beta platelet sheets are two types. One is a parallel, and the second one is the anti-parallel. And in the parallel beta platelet sheet, if the two polypeptide chains run side by side in the same direction, you can also notice here that this is C terminus and the C terminus, this is N terminus and this N terminus. That means both the polypeptide chains are running in the same direction. Then it is known as the parallel beta platelet sheet. Whereas if they run in opposite direction, C to N, and whereas the N to C, and but these two are connected by the interchain hydrogen bonds between the carbonyl oxygen of the one polypeptide and the amide hydrogen of the another polypeptide. And so that means the beta platelet sheets are stabilized by the interchain hydrogen bonds. And you can also see here that in case of parallel sheet, there is a distortion in the hydrogen bonding compared to the anti-parallel. That's why anti-parallel beta platelet sheets are more stable compared to the parallel beta platelet sheets. And in addition to this, as I already said, that secondary structures are either ordered or the disordered. Order, what we have said, is alpha helix and the beta platelet sheet. And we also have the disordered secondary structure or the disordered conformation. And under this, we have the beta turn or the beta bend. And these beta turn or the beta bend, it is also known as the hairpin, and it is composed of the 4 amino acids, mainly glycine and the serine, aspartic acid, and the proline. And it is mainly involved where the direction of the polypeptide chain has to be changed, and this region, the beta turn or the beta bends are present. In addition to this, there are some super secondary structures. These super secondary structures are found in it's composed of more than one secondary structure, and you can also see here all these super secondary structures are beta alpha, beta unit, and Greek key, beta mender, beta barrel. And you can also see here that the beta alpha beta subunit consists of the two parallel beta sheets are connected by the alpha helix, and here the Greek key is composed of several anti-parallel beta strands are folded, and whereas the beta mender, and it is also consists of the 5 beta sheets connected by the several reverse turns, and the beta barrel, and it has many parallel beta platelets. It forms a pore-like structure, mainly, especially the porins. The beta barrel is present. These porins acts as a channels. And coming to the domain, we know that when we say that when we talk about the protein, we say that the protein has the 3 domains or 2 domains, and the domain is nothing but it is the portion of the protein that has its own function. We will discuss this one in the tertiary structure. And coming to we know the triple helix. We know that the collagen has 3 polypeptide left-handed helices coiled in a right-handed fashion. So this is it has a triple helix, and it is a super secondary structure. And the detailed structure of the collagen we will discuss in the later. And this is about the secondary structure. Coming to the tertiary structure. The tertiary structure, as I already said, that it is the 3-dimensional structure of the protein, and it is composed of several alpha helices, beta sheets, beta turns, and the random coil conformations. And these tertiary structure is stabilized both covalent as well as the non-covalent interaction, non-covalent bonds, mainly the covalent bonds, mainly disulfide bond, whereas the non-covalent interactions are hydrophobic, ionic, and hydrogen, and as well as the van der Waals forces. So that means when the polypeptide chain folds into its three-dimensional shape by the association of many alpha helices, beta sheets, beta turns, and the motifs, the three-dimensional structure is formed. And when the protein folds into its three-dimensional shape, and most of the polar amino acids stay on the surface, because in the cellular environment, the cytoplasm is aqueous in nature, and so therefore the polar amino acids stay on the surface, whereas the non-polar amino acids are buried inside the protein. That means when the protein folds into three-dimensional shape by the association of several alpha helices and beta sheets, always the polar amino acids stay on the surface, like non-polar amino acids like valine, isoleucine, and other non-polar amino acids are buried inside, because they tend to escape in interacting with the aqueous water present in the cytoplasm. So that means the conclusion is that when the protein folds into three-dimensional shape, and the protein folds into three-dimensional shape, and the protein is surrounded by a layer of water molecules, and this layer is also known as the hydration layer or the solvation layer. And so therefore, in order to interact the amino acid side chains with the water, so only the polar amino acids can present on the surface, whereas the non-polar amino acids are buried inside the protein. So this, you will understand why the non-polar amino acids are buried, because cytoplasm is aqueous in nature. Obviously, the non-polar amino acid cannot tend to be on the surface. And so this is a structure of the three-dimensional structure of the various proteins. You can amylase and the pepsin and the trypsin, you can see here. And this is the ribonuclease. It has many amino acids, you can see here. And so then coming to the domain, as I already said, that we in many proteins that are present in the cell are the multimeric proteins, and they have that more than one subunit. And in addition to this, we generally say that the protein has three domains or the four domains or the ATP binding domain and so on. That means domain means it is the part of a protein which has its own function. So that is called the domain. For example, pyruvate kinase with three domains, that means each domain has its own function. So that means a part of the protein which has its own function is known as the domain. So this is the structure of the three-dimensional structure of the protein. Now coming to the quaternary structure, quaternary structure of the protein. And the quaternary structure, as I said, that association of two or more polypeptide chains is known as the structure. And in this quaternary structure, so each subunit is also known as the protomer or the monomer or the subunit, and that means it is composed of quaternary structure is nothing but the association of many subunits. As we have already said that the hemoglobin, it has the two alpha subunit and two beta subunit, all are connected by various non-covalent interactions. Therefore, the hemoglobin exists as a quaternary structure, and this quaternary structure is stabilized by, as I already said, that both many non-covalent interactions and also the covalent bond, meaning the disulfide bridge, like disulfide bridges. So this is about the quaternary structure. So when the protein is denatured either at the extreme alkaline pH or the acidic pH or the high temperature, or the protein loses its three-dimensional structure, as a result it becomes linear, and that means the loss of three-dimensional structure of the protein is known as the denaturation. And so this is about you can also see here when this protein is denatured and it loses its three-dimensional structure. So this is about the protein structure. In summary, what I wanted to say. in the summary, what we have discussed in today's class is that we have discussed about the peptide bond, and we said that the peptide bond is a amide bond, and it has the 40% double bond character because of the resonance of carbonyl pi electrons and the lone pair on the amide nitrogen. Then we discussed about the various physiologically important such as thyrotropin releasing hormone and glutathione and the chemotactic peptide, vasopressin, angiotensin, bradykinin, as well as the the enkephalin and so on. These are all physiologically important peptides. And then we discussed about uh, the properties of proteins, and we have already said that proteins have various functions. They have the storage function, transport function, defense mechanism, contractile function, and so on. And some toxins are also proteins in nature. Then we discussed about the charge properties of the protein. And in charge properties of the protein, as I already said, that the charge of the protein is dictated by the surrounding pH of the surroundings. So it is similar to the charge properties of the amino acids. And then we discussed about the isoelectric point. The isoelectric point is the pH at which the net charge on the protein is 0, is the isoelectric point. And the isoelectric point depends on the composition of the amino acid residues present in a protein. For example, if the protein has more of basic amino acid, its pA value would be on the basic side. If it has more of acidic amino acids, and its pA value would be on the acidic side, and so on. That means the pA value is dictated by the amino acid composition. And all of you need to understand that when the protein is formed with the amino acids, and you should only remember that only amino group of the amino terminal amino acid and the carboxyl group of the carboxyl terminal amino acid are only free, and remaining all amino terminal and the carboxyl terminals are involved in the peptide bond formation. Only the side chains, if it is only the side chains, only the side chains, only the side chain groups are only the, if it is acidic amino acid, it has one extra carboxyl group. If it is basic amino acid, it has one extra either amino group or the guanido group, and so on. So that means depending upon the pH, these side chain groups, either they proteinate or they deproteinate, that in turn dictates the stability of the polypeptide chain. So that's why, so the pA of the protein depends on the amino acid composition, and so on. Then after that we discussed about that the structure of the protein, and the protein structure of the protein, and the protein structure is divided into four levels. That is the primary, secondary, tertiary, and the quaternary structure. And the primary structure refers to the sequence of amino acids present in a polypeptide chain, that is known as the primary structure. And this primary structure is mainly stabilized by the peptide bonds. In case of insulin, it is also stabilized by the disulfide bonds. So that is the primary structure. Coming to the secondary structure, when the polypeptide chain has many amino acids, such a lengthy linear polypeptide chain is highly unstable, and this polypeptide chain tends to form ordered, or they tends to fold into stable conformations, and these folded structures are known as the secondary structures. We have the two types of these. Folding can take place in the ordered fashion or the disordered fashion. There we have the two types of ordered secondary structures. One is alpha helix. Second one is the beta platelet sheets. These beta platelet sheets are either parallel or the anti-parallel. Alpha helix is stabilized by the intrachain hydrogen bonding, whereas the beta sheets are stabilized by the intrachain hydrogen bonding. and we have already discussed about the certain characteristic features of the alpha helix. In the sense, each turn has the 3.6 amino acids per turn, and the R groups are always present on the outside, and the every amino acid, every carbonyl oxygen, and the amide hydrogen are involved in the intrachain hydrogen bonding with the fourth amino acid. Therefore, the alpha helix has the extensive rigidity and stability, and certain amino acids stabilizes the alpha helix, certain amino acid destabilizes the alpha helix, and then we discussed about the right-handed alpha helix is more stable than the left-handed alpha helix. Then we discussed about the beta platelet sheet. In the beta platelet sheet, the parallel beta platelet sheets compared to the parallel beta platelet sheets, anti-parallel beta platelet sheets are more stable, in the sense they have the less distortion between the hydrogen bonding between the two chains compared to the parallel beta platelet sheets. And these beta platelet sheets are present in many proteins. And then we discussed about the tertiary structure. Tertiary structure is composed of many alpha, many beta platelet sheets, many super secondary super secondary structures, and beta turns and the beta bends. And this is the tertiary structure is nothing but the three-dimensional structure, and this three-dimensional structure is stabilized by the both covalent as well as the non-covalent interactions. And these covalent interactions are mainly the disulfide bonds, and the non-covalent interactions are the hydrogen bonding, ionic interactions, and the hydrophobic as well as the covalent forces. And when the protein folds into the three-dimensional structure in the cell, and the protein is surrounded by a layer of water molecules, and this layer is known as the hydration layer or the solvation layer. And since the cellular environment is aqueous, when the protein folds into a definite shape or definite three-dimensional structure, most of the polar amino acids stay on the outer surface because they can interact with the water molecules, whereas the non-polar amino acids stay in the inside because they doesn't favor in interacting with the water. And this is about the this three-dimensional structure of the protein is very, very important for the protein to function. And when the protein loses its three-dimensional structure, it can no more exhibit its function. And then coming to the quaternary structure, we already said that the quaternary structure refers to the association of two or more peptide chain by the non-covalent as well as the covalent interactions. And in these each subunit, each monomer is known as the protomer of the subunit, and they are held together by the various non-covalent interactions and also the disulfide bonds. And this quaternary structure, many oligomeric proteins present in the quaternary structure like hemoglobin and other oligomeric proteins, they present in the quaternary structure and quaternary structure. Then we discussed about the denaturation of the protein. When the protein is subjected to the alkali or the acid or the heat treatment, the three-dimensional structure would be lost. As a result, the protein becomes linearized, and this loss of the three-dimensional structure is known as the denaturation. And so this is about the protein structure. So just take 5 more minutes to explain why what is the Ramachandran plot or the Ramachandran diagram. And this is as I already said, that the peptide bond has the partial double bond character, and we have already said that C double bond O NH, this is the peptide bond, and this side it is C alpha, and again this side C alpha. It has the side chains. As we already said that in a peptide bond, the R groups is always present on the trans configuration. In the sense, they are present in the, you can see here, if it is one R group is here, another R group is on opposite side. And in this peptide bond, as I already said that the peptide bond has certain characteristic features. In the sense, it has the partial double bond character. In the sense, it has a 40% double bond character. And this double bond character is because of the resonance of the carbonyl pi electrons and also the lone pair on the amide nitrogen. And so since the peptide bond has the double bond character, and this double bond, so this is the C double bond O NH, and this is the double bond character. This double bond, since it has a 40% double bond character, and this double bond doesn't allow the rotation of the rotation of the atoms. In the sense, rotation is not possible around the peptide bond because it is a double bond, it doesn't allow the rotation, whereas the rotation is possible on both sides of the peptide bond. One side it is known as the C alpha and C double bond O. Another side it is known as the C alpha and the NH. So, and if you see here that if the C alpha and C alpha C double bond O, rotation between the C alpha and the C double bond O is known as the psi, and the rotation between the NH and C alpha is known as the pi. That means when you take any protein, when you take any polypeptide chain, C double bond O, NH peptide bond, so rotation is not possible. Again, this is peptide bond, again rotation is not possible. This is peptide bond, rotation is not possible, and this is peptide bond, rotation is not possible. So rotation is possible only on both sides of the peptide bond. For example, here this side it is possible, and this side is also possible. So the C alpha and the C alpha and the C double bond O, the rotation between the C alpha and the C double bond O is known as the psi, and the rotation between the C alpha and the NH is known as the pi. So what G.N. Ramachandran has measured the psi and pi angles for various peptides using models, and he has constructed a map that is also known as the Ramachandran diagram or the Ramachandran map, and it is also known as the pi psi plot or the pi psi map. And so when he has measured the pi and psi angles for various peptides, they have constructed the map between the pi and the psi. And what he observed is that and almost the psi and pi angles for most of the proteins are very minimal. Only that means in this entire map, only 10% of the psi and pi combinations are only seen in the proteins, and they are all falling in a particular region, and he named it as the shaded zone. So you can also see here in this entire map, when he has constructed the pi and psi angles for various proteins using models, and all these pi and psi combinations are falling only on a particular region of the protein. In the sense, each dot in this map represents the combination of one psi and pi angle, and you can see here the entire map is empty. That means in a protein, only particular psi and pi, in a protein, the polypeptide can exist a stable conformation at only particular psi and pi angle. And whereas if we synthesize any protein, and if it is pi and psi angles are falling outside the shaded zone here, so that means this protein doesn't exist a stable conformation. So that means this Ramachandran has developed this map, and it is useful to visualize dihedral angles psi against pi of amino acid residues in a protein, and it also shows the possible conformations of the psi and pi angles for a polypeptide chain. So as I already said, that the peptide bond has a 40% of the double bond character, and this double bond doesn't allow the rotation. It restricts the rotation, as rotation is possible only on both sides of the double bond. And so G.N. Ramachandran has measured psi and pi angles for the various peptides, and he has constructed a map. And in this map, only 10% of the 10% of the in this map, only 10% of the map has only the shaded zone that indicates the allowed psi and pi combinations, whereas the remaining region is forbidden angles of the psi and pi. So this is about the Ramachandran map. It is useful to understand the prediction of the protein, whether it can take up the parallel or the anti-parallel beta sheet or some other alpha helix and so on. You can also notice here that the psi and pi combination, the psi and pi values for different proteins are different. You can see here for alpha helix, right-handed alpha helix, the pi value is minus 57 degrees and the psi value is minus 47. For parallel beta platelet sheet, it is pi value is minus 119, the psi value is 113, and the anti-parallel minus 139, 130. That means if you take any polypeptide, when you measure the psi and pi throughout the polypeptide, it has only, if it is alpha right hand alpha helix, it has only pi value minus 57. minus 40. That's why, since it has the same pi and psi values, that's why they form the ordered secondary structure. So that means the when the rotation all the in a polypeptide chain, the atoms cannot rotate beyond the certain degrees, because when the atoms come closer, you would also notice that they have the steric clash. You can see here the side chain, one amino acid, another chain of another amino acid. So when they rotate beyond certain degrees, they have the steric clash, because the electron cloud on the one atom has the steric clash with the electron cloud of the another atom. So that means all pi all pi and psi combinations are not possible. Only few pi and psi combinations are only seen in the proteins. That's why the entire the Ramachandran map, only 10% of the psi and pi combinations are only seen, and the remaining pi and psi combinations are not seen in this one. So this is about the Ramachandran diagram. It is already given in the material in the Leninger. You can also go through that material for better understanding.